I'll drop the light so that everybody starts quieting down.
Okay.
And then you can get going.
Sounds good.
All right. I'll be right back there.
People still coming in. So welcome and thank you for coming to the final STEM lecture series for spring of 2018. And first, a thank you to Dr. Justin Topp and Amanda Finnegan for their work putting this lecture series together. And thanks for giving me the opportunity to invite Dr. Hans Dam to be a part of this lecture series. So Dr. Dam is a biological oceanographer from the University of Connecticut. He earned a BS in oceanography from the University of Washington and an MS and a PhD from SUNY Stony Brook in marine science and coastal oceanography. He's been a professor at UConn since 1991, and during these 27 years, he has had a very distinguished career, receiving numerous grants to do research all over the world and publishing numerous peer-reviewed papers. He's also received many awards, including being made a fellow of the American Association for the Advancement of Science, a sustaining fellow of the Association for the Society of Limnology and Oceanography, a member of the Connecticut Academies of both Arts and Sciences and Science and Engineering, and he also received an NSF Ocean Science Career Award. But even with all these honors, Hans claims that one of his proudest accomplishments is his training of graduate students. He has trained 20 or so over the years, and I was his first one. So I started my master's in oceanography in 1990, right before Hans arrived at UConn. And Hans was very instrumental in my own training and the trajectory of my career. It's a wonder I actually made it through, considering all the glassware I broke and all the other mistakes I made in the lab and in the field. But Hans was very patient with me, and he only made me cry a few times. But he taught me a lot about how to do research and how to analyze and present data, and among other things. But much of what I teach during thesis is stuff I learned directly from Hans. So he also took me to my first professional conference on my first research cruise, which we were just talking about, was 30 days sitting in the middle of the equatorial Pacific with no land for hundreds of miles in all directions, with very sleep deprived. But so as you can imagine, all of this had a tremendous effect on me both professionally and personally. So I'm so happy that Hans was able to come to Endicott and be part of this lecture series. And I hope that you'll enjoy his talk entitled The Arms Race Between Predator and Dangerous Prey in the Ocean. Please join me in welcoming Hans Dam.
Hey. Can you hear me okay? Great. Thank you so much. It's really an honor and a pleasure to be here. Thank you for the nice introduction, Mary. I have many stories about Mary, and she has many about me. We'll save those for dinner. But particularly the fact that Mary was my very first graduate student. So it was baptism by fire on both ends. And she survived me, and I survived Mary. And but I count Mary as a dear friend. She was in my 50th birthday party. Not that, well, actually it's a while back now. And I'm delighted to be here. So it's really an honor. And this talk has given me an opportunity to sort of rethink a lot of the work I've been doing in the past. So let me just get started here. Oops. Oh. No, I'm good. I'm good. Okay. Okay, so I changed the title just slightly, just to emphasize that I'm talking about the plankton. Okay, so the arms races between predators and dangerous prey in the plankton. And if you are interested in asking me questions, if you're too shy to ask questions here, you can always reach me by email. There's my email address, and I'm happy to talk to anybody. Okay. And feel free to ask questions in the middle. If you want something clarified, let me know. If it's going to be a debate, we'll wait till the question-answer period. Oh, okay, so the first thing is I want to thank Mary for the wonderful present. I just got a present 2 hours ago. Went back to my hotel room, opened it, and I said, okay, yeah, look at this, fantastic. So thank you for the memento. And a cap is always great. And I can then tell people about Endicott College and about the wonderful fighting gulls. Okay, all right.
Full when she gave it to you?
Yeah, after dinner. Yeah, okay. Okay, and before I forget, right, because I have poor manners generally. Before I forget, I want to remind everybody that what I'm talking about today is a joint effort with many people, right? And none of that would have been possible without the generous supports of many funding agencies and of all my students and collaborators. And you can see we started this work about 15 years ago. I was considerably younger, no gray hair at the time, as you can see here. And these were the very first two students who worked on the problem. And this is Dave Avery, who is now an associate professor at Maine Maritime Academy. He was a postdoc in my lab. Tina Samt, who got her PhD. Kihon Park, who is now finishing. Sir Burris, who got her PhD there. Li Hua Chen, who got a PhD. Michael Finiguera, who also got a PhD and is now a professor at UConn. And then the faculty collaborators, including people from the University of Florida. So I don't want anybody to think that I did this on my own. Mostly my job was to shake my head and said, yes, okay, good idea, let's do this. Okay. Oh, thank you. All right, arm races. So I hope, I think most of you are too young to remember this, but some of you will recognize these two characters here. Anybody have any idea who these folks are in the picture? Anybody that doesn't have gray hair? Kennedy and? Yeah, Nikita Khrushchev, right? So this was probably, I cannot, there's no credit here because I can't quite figure out where this came from. I did a search and this is what I found, but I don't know who made it, but I have a feeling that this was done right after the Cuban Missile Crisis, right? And it had to be done by an American cartoonist because Khrushchev is the one who is sweating. The Russian version would have Kennedy sweating, okay? And it's interesting because you can see in this picture that there's a mano a mano here, right, these guys. And they're both sitting on hydrogen bombs. And as you can see, Kennedy has his finger over the button that would detonate the hydrogen bomb for the Russians. And Nikita Khrushchev, on the other hand, has the same thing, right? So there's these two guys who are in a battle that could be mutually destructive. And this is what we tend to think of as arm races. There's other examples that you can think of, but actually on the way here, driving from Connecticut, so quite a few cops with their radar guns, right? So there's another kind of arm races that you can think of, which is the cop wants to make sure that you don't speed too much, right? And they have their radar guns to catch you. And in the meantime, we've developed all this fancy technology to avoid the cops. So Waze was telling me, police car ahead, right? And I would be able to slow down, right? So this is another kind of arm races that you can think of. But arm races have an interesting characteristic, which is, here's another example that it's a little bit more current, quite current, as a matter of fact, given what the situation is. So you can recognize the fellow here on the left is Vladimir Putin, looks awfully like Vladimir Putin, right? And he's flexing and telling, hey, how about this? And then NATO said, well, what about this, right? Oh, yeah, and you can see that they're getting bigger and bigger. The muscles are getting bigger and bigger, right? So one characteristic of arm races is that there's this idea of escalation, right? There's a tit for tat, and there's therefore an escalation, and that's where the arm races idea comes from. And just to give you an idea, there was a real arms race, and there still is some, but this is a number of warheads by the U.S. and back then the USSR and Russia, and you can see that there was a tremendous buildup of armament by the U.S. that then was matched by the USSR, but there was at some point a decline. Okay, so that's the possible third characteristic of arm races. The first for that is its antagonistic relationship, right? There's a tit for tat. The second is that there's idea of escalation. I hit you back, I hit you harder, I hit you harder. And the third is that there may be a cause, right? So what is this idea of cause? Well, there are many ways to think about that. One is that it may actually be, in terms of resources, it must make us a lot of money to build all this armament. And in fact, the thesis has been made that many an empire has collapsed because their army has grown way too big, and the needs of the nation have been neglected, leading to social collapse, okay? So that's one possibility. The other possibility, of course, is what is called opportunity cost, which means, well, if I'm busy engaging in this arm race, I'm missing the opportunity to do other things, like building universities and so forth, right? So the direct cost of the arm race is an indirect cost as well. So the same thing may happen with biological systems, but there are slight differences. So the first thing is that the players in the arm races are not individuals, but they're lineages. We're still probably thinking of about groups of individuals, populations, all right? The second thing is that the relevant time scales, because arm races involve natural selection, is generations, right? This is the time scale that matters to the players, because changes occur through generations. The third is that to be a true arm race, the relationship has to be symmetrical. The war has to be symmetrical. So many of the things that you hear about being arm races are not, and I'll give you an example in a bit. The fourth is that to make sense of them, you have to, the right currency to use is fitness, right? That's how you end up measuring things. And one thing that seems to be characteristic of arm races is that they actually include dangerous prey, all right? And that is because of the issue of the symmetrical selection. So you may have seen, a lot of people think of this as an arm race. So we have a gazelle here, and we have a ferocious predator, right? A cheetah, or that looks like a cheetah maybe. And they tend to think, oh, they're involved in an arms race because if the cheetah gets faster, right, the gazelle gets faster. And if the gazelle gets faster, the cheetah must get faster. But that's coevolution, but it's not really an arms race because it's not symmetrical. The reason for that is that if the cheetah does not catch the gazelle, there's no big consequence. The cheetah goes hungry, but it can go after slower prey. If the gazelle, on the other hand, fails to escape the cheetah, there is a huge price to pay, which is death, right? So the selection is for the prey. There's relatively little selection for the predator in this case. So that's really does not qualify as an arms race. But when you have dangerous prey and predators, it is a symmetrical relationship. The classical example for land is the case of the garter snake and the poisonous newt. So out in California, this fellow, Edmund Brody has made a very distinguished career studying this problem. The newt produces a toxin called tetrodotoxin. And that's a toxin that you find in pufferfish. That's where the name comes from, the genus of pufferfish. So I don't think you will do this, but if you're in Japan, you can actually eat poisonous fish. And it is a sort of a thrill to do. It's expensive, because you have to have very trained chefs to do this. And you know when you're eating it, because you get a nice little tingling in your lips. A little too much will kill you, because tetrodotoxin is a very potent neurotoxin. And the way it works, like the one toxin I'm going to talk about later, is that it blocks your sodium channels. And you may remember that sodium channels are important because that's how neurotransmission occurs between cells. So if you stop neurotransmission, right, what you're going to do is you're going to paralyze muscles. And then if you paralyze enough of those, your diaphragm will stop working, and you will asphyxiate. So it's a pretty horrible death. All right. So you want to be careful. So the idea with this guy is that these snakes can eat these poisonous newts without dying. But not always. There is variation in the lineages of snakes, right? So let me give you an example of how that may work. And this is sort of a summary figure for that, is that if you plot newt toxicity of the prey here versus what is called the tolerance or the resistance of the predator, there's a very tight correlation between the two. And the way they measure the tolerance of the snake here is by how fast it can crawl, right? So that's sort of a measure of fitness for the predator here, right? So you can see this idea of tit for tat and escalation, because where the toxicity of the prey is low, the tolerance or the resistance of the predator is low. And when the toxicity is very high, the resistance of the predator is very high, which suggested this idea of coevolution and this tit for tat escalation. And that leads to this idea, if you may have heard this, of red queen dynamics, right? So how many of you have read Alice in Wonderland? Okay. So remember that one of the laments from Alice is that she had to do all the running in the world to keep in place. So Alice was chasing the queen, but they went nowhere, even though they were running really, really fast. So this idea of the predator and the prey catching up with each other, but going nowhere, okay? So you might hear that term later, all right? So question is, do we have arm races in the plankton? Because you can think of how this coevolution can happen in land, where you have the localized population with specific habitats, right? And where there could be selection, natural selection going on. And then you can have these separate populations. But the ocean is a fluid. And the plankton, by definition, are drifting, right? They move with the currents. And then, there's this controversy as to, can you have strong selection because there's so much gene flow in the ocean? Thankfully, we have estuaries where we can test this. So in the open ocean, I don't know how it would be very hard to test an ocean. But in estuaries, there are sort of localized population where the gene flow may be less, and then you can perhaps study. So the question is, do we have them? And it's also difficult. Well, I just want to show this incredible picture of plankton. And if you want to learn more about it, there is a wonderful website called the Plankton Chronicles. If you just want to, one day instead of watching Netflix, go watch this. There's some incredible videos. There's cool music, and you can see these plants and animals doing all kinds of bizarre things. So go there, take a look. And these guys are organized. These are all the primary producers here, all the phytoplankton, and then they move on to the consumers here, right? The thing about plankton is that they're also very tiny. So the scale here is in micrometers, right? So this is 0.2 microns, 2 microns, and it's a log scale. 1,000 microns is 1 millimeter, and that's about the size of a rice of grain, all right? And you can see here that there's the copepod here. This is a metazoan, right? All these guys are protists here. They're single-cell organisms, right? So the arms race that I'm going to talk about is between one of these copepods, and here's a picture of one of them, one here and one here, right? So they're like cousins of shrimp, tiny, but somewhat related to that. So it's hard to study them because they're so tiny to begin with. It's hard to grow them in the lab. So a lot of people don't like to do this. Only masochists like me that have decided, oh, we can try this, okay? All right, so for the students, what do I want you to walk away from this lecture? Okay, the first thing is that, okay, it's a cool subject. I mean, come on, arm races, right? So it's fascinating from the evolutionary point of view if you're interested in evolution, right? There is evidence. I'm going to show evidence that they do indeed take place in the ocean, right? There's very interesting and societally important implications to the arm races. I'll make a case for that, all right? And the other thing is this is a STEM lecture, and my understanding is that you're really interested in sort of interdisciplinary, multidisciplinary approaches. So it is my view that the really cool problems in biology these days and in oceanography are all very interdisciplinary. So to address this problem, you need to know a bit of everything. And chances are you won't know all of that, but if you're smart, you'll just hook up with the right people who know about it, okay? So you need to know some ecology, some evolution, some molecular biology, some physiology and neurobiology, and some toxicology, okay? All right, so these are the players. There's a picture of the copepod, and then there's a picture of a toxic dinoflagellate. I'm just going to show you more detail about them. The toxic dinoflagellate is in the genus Alexandrium, and here is a blow-up of them. Here's 2 cells, right? And they're called dinoflagellates because they have a flagellum. They actually have 2, these guys, right? And you have them right out here. Probably not now, but you might have them in the summertime out here. So this particular species is found from New Brunswick, Canada, all the way, the southernmost end is about Long Island Sound, where I work. All right. Like the newt, they also produce a toxin called saxitoxin, which is a very potent neurotoxin, right? Blocks sodium channels, cause paralysis. And it's extremely potent. I don't know if you ever heard of the poison curare, that the South American Indians, when you see the movies where the Indians go with the dart gun and hits a guy, the white guy, and he dies, right away. Well, this stuff is 1,000 times more potent than curare. So you want to stay away from it. You don't want to eat it. Okay. And then the copepod species is called Acarcia hudsonica, sort of a generic copepod we have. It's all over the Northeast. Okay. So just to show you that I'm not lying, I'm going to show you a little video of how. What happened here? It's not showing. There's supposed to be a player. This one?
No, it's actually flat.
Oh, no. One. I may have to get out of this present. Oh, there it is. All right, so this is a short clip, right? So you see the copepod. It's in a petri dish, and I have taken a video under a dissecting scope like you would have in your lab. And the little dots you see are the dinoflagellates, and they will be moving, right? Because they have the flagellum, they can swim around. And you see the copepod eating them. And then I'm going to show, later you'll see the same copepod 24 hours later and see what happens. Oh, there we go. So there is eating happily, munchies, eating. 24 hours later, there it is. See how slow he is? And he's swimming backwards. He's completely intoxicated. Right? So yeah, I'll play it again. So, ooh, so delicious. Yeah. This is about 40x. Right. So the copepod is 1 millimeter or so, and the dinoflagellate is about 20 microns or so. Right. So that's characteristic, right? So what's happened is the copepod has eaten all the toxic algae, right? And then it's poisoned. It's not dead, but it's actually pretty intoxicated, and it becomes lethargic. The swimming behavior becomes erratic. Of course, if they eat enough of that, they will die. But in this case, it didn't die. Okay. This shouldn't have done that. Hold on. Yeah. Oh, no. Oh, sorry. Sorry, there we go. Okay. So who cares, right? Why? I mean, okay, yeah, there's this arm races, blah, blah, it's interesting, professor, yeah. There are practical implications to the arm races here, right? So here's the figure on the left here shows what are called events of paralytic shellfish poisoning, right? So the reason you care about the toxin is not because it affects the copepod. I use the copepod because I like them. And I use the copepod because they have short generation times, and I can manipulate them in the lab. Of course, the copepods are also the most abundant animals in the planet. If you didn't know that, there's your cocktail factoid for you, right? There are more copepods than insects, right? And of course, they are the main food source for fish. So no copepods, no salmon, no cod, no sushi, none of that stuff. All right. So, but the reason you care is because of shellfish poisoning. So I don't know if many of you do some clamming around here. Sometimes the clam beds are closed. You cannot go there because the clams are loaded with taxitoxin because they've eaten the cells, right? The clams filter the water, and then they're loaded with cells. If you eat the clam, you will get very sick or you could die, all right? The first case of PSP that we know of was out on the East Coast. So you can see in the 1970s, most of them were on the on the West Coast. And back in the 1700s, there is a record from Captain Cook. Several of his sailors went to an island off Vancouver, right, and didn't come back. And when they went to look for them, they found them all dead. And they found them they had eaten some clams. So chances are that they poisoned themselves, all right? And nowadays, usually people who die from eating contaminated clams is because they're tourists that are not in the wild and decide, oh, there's some clams and some oysters and scallops and so forth, and they eat them, and then they find them days later dead from eating that. But the point is, if you compare 1970 to near now, right, there's a huge change in the number of these PSP events. There's way many more nowadays, right? So the question is, okay, what does that mean? Why are they proliferating? There are many reasons, we won't get into it. But the fact is now they're very common everywhere. And that means then we have to worry about them in terms of toxin transfer, right, in the food web, which disrupt these food webs. And it's also very expensive. When these climbing beds are closed, people can work, right? So there are many millions of dollars lost to the economy, the sea economy of New England, for example. And the other is that you might wonder, right, because of these arms races, is whether the grazers themselves can keep the plants in check or not. All right, so some implications right there for everybody. All right, so how do we know that there is this co-adaptation between the predator and the prey, right? So we decided to tackle that question a while back, and we knew one thing is that this is a copepod, not to scale. Okay, that's a picture of a copepod. This is an idealized picture of a copepod. All right, and we knew that this is Long Island Sound here. Where you guys are around here somewhere? No, this is Cape Cod, so you're up here somewhere, right? So from Massachusetts and up, the copepod and the toxic algae co-inhabit. They're together. And then as you go down south, you find less and less of them, right? And if you go south of Connecticut, you don't find any more of the toxic algae. That's their limit, right? Okay, so we knew that, and we said, okay, so we said, okay. The other thing we knew is that the blooms of the toxic algae are really potent up north in Canada, right? And they happen, we know that they've been around for a long time, and they happen very often. So we said, okay, we have a nice gradient in the duration of the blooms, their toxicity, and how long they've been around. Maybe there has been natural selection for the grazers in the northern waters to become tolerant to the toxic prey. And just to make the case some more, here's the toxicity of those cells as we go. This is Connecticut is 41 degrees or so, right? And then this is 46 degrees. That's like New Brunswick, Canada. And you can see that there's a strong gradient in the potency of the poison, right? So just like the new situation, then you might expect to see that the tolerance of the predator follows this gradient. Okay. So how do you test for that, right? So that it said, okay, why don't we just go to all these places? We means in the royal sense, meaning my student at the time, Sean Collin, who is now a professor at Roger Williams. He said, I'll just go, I'll get in the car, and I'll go and collect populations all over, right? But you can't just bring them and then do an experiment right away because you don't know whether the conditions were the same, right? So what if, we're thinking, okay, these guys are going to be less tolerant than the ones in Maine, but what if they had a lousy season and they were really weak and skinny and, so that gets confounding, right? So the way to do the experiment is to say, okay, let's bring them home. Let's put them in the Hilton, right? Bring them to the lab. Here's all the populations. You grow them, you put them in these big buckets and give them lots of food and you treat them really well. And you do that for several generations. So you remove all those carryover effects, right? So remember, and of course, under non-toxic food, you give them good food. You're not going to poison them. So remember then that any changes, any variation, phenotypic trait you see has three components. One is the genetic component, one is environmental component, and then there is the interaction of those two components, right? And then by keeping them in these beautiful conditions and standardized, we eliminate the environmental effect, and therefore we eliminate the interaction effect, and then we have, if we see any difference, they have to be genetic, okay? So what do we see? What I'm going to show you is results from a bunch of papers from a few years ago. So the first one is effect on reproduction, right? Because that's a proxy of fitness. So this is what happens when you give them non-toxic prey. You give them some nice food, right? So what do you see? These are all populations from different places, right? New Brunswick, Maine, Massachusetts, Connecticut, and New Jersey. The ones farther south, New Jersey and Connecticut, are in the warm color, the red and orange. And then to the north, they're in the cool colors. And you cannot really see any statistical difference among them. They all could be essentially the same line, right? But when you give them the toxic food, you see that they split into two groups. And it's a very striking difference, a significant difference, right? So the guys from the south that do not see the toxic blooms frequently, right? They take a big hit. Their reproductive rate really suffers. Whereas the guys who cohabit with the toxic algae do quite well, right? So that's the first evidence that there is some sort of adaptation in the animals that see this toxic blooms often. Okay, if you now, what I'm going to show you now is I'm just going to take two of those populations. I'm going to take the northernmost, Maine, and compare it to the southernmost, New Jersey. And the one from New Jersey is going to be the control, right? If you now compare their survival from being born to when they become adults, then you can see we have a control food, which is the good food, it's not toxic, and then the treatment is the toxic food, all right? And you can see that the animals from New Jersey, again, survive much worse compared to in the toxic food relative to the control food, but the ones from Maine, they do just as well. They do in both of them, okay? So that's another piece of evidence, right? And then when they become adults, if you repeat those egg production experiments that I showed earlier, you see that the ones from New Jersey, they take a big drop in their egg production, whereas the one from Maine do not. There's no difference between the foods, right? All right, so that's all consistent with this idea of adaptation of the populations that cohabit with the toxic algae. And then, if you remember, the fitness is a product of survivorship and reproduction. So you can combine those two to come out with a quantitative measurement of fitness, which I'm calling lambda here, right? And that is the net population growth rate. What does that mean? It means how many individuals, each individual in the population is going to leave behind, right? So anything greater than 1, that means that the population can grow. All right. So these guys here produce 1.6 individual each, and this produce 135. So in the New Jersey animals, right, the control food is a good one, the treatment food is a poisonous food. You see that there is a decrease, there is a significant decrease in their fitness, whereas the one from Maine do not experience that decrease, right? So there's a fitness tax in the animals that are not historically exposed, but there is no fitness tax in the ones that have co-evolved with the toxic algae. Okay. So now if we go back to the arm races in the ocean and on land, remember the picture of the snake eating the newt, right? So there's a gradient of toxicity, and you see a gradient of resistance or tolerance. So you would expect the same thing here, right, in our gradient of toxicity. And you do see that, right? Where we have low toxicity, we have low reproduction, low survival. Where we have high toxicity, we have high reproduction, high survival, because of this co-adaptation. Okay. All right. How am I doing for time? Okay. Oh, okay. I had to talk too much. I'll talk less. I get excited. That's the problem. Excitable boy. So what about the prey? That's the prey, right? Remember, it's tit for tat. So we've shown an adaptation of the predator, but does the prey adapt to the threat of the predator? And the answer is yes. So this, again, work by one of my former students. What Tina Senf was able to show is if you just put in a jar a bunch of Alexandrium cells, right? One control, there's no animals. The treatment, you put an animal in there, and you measure the amount of toxin per cell, there's no change in the control. Okay? These guys, the toxicity doesn't change. You bring the animal in there, and the toxicity goes crazy immediately. So it's an inducible defense right away. Okay. And then they said, okay, but that's just for copepods. So I said, no. So we did a bunch of other experiments. It was slightly different. In this case, when you're working with little animals and big animals, it gets complicated because the big animals can eat the food too fast. So we said, okay, let's do a different kind of assay where we just, it's sort of the presence of the predator. It's just the threat of the predator, right? So we measure the toxin in the cells that are separated from the predator by a mesh. So the ones here down below, these guys here, right, they're never eaten by the predator because this mesh keeps them separated. So what we did is we put the predator there, we measured the toxin content of these guys here and relative to a control, right? And then this shows here the increase in toxin content and the toxicity of the cells, of these cells that were exposed to the predator. And you can see, and all of these guys in the bottom here are different kinds of predators. So they're copepods, they're bivalves, they are tunicates, and so forth, right? So regardless of which kind of predator you put in there, the mere presence of the predator tells the algae, start cranking up the toxins. And then if you measure how many cells were ingested in those experiments that I just showed you versus the toxin production, you see this relationship. Now, this is a log scale here, right? So this relationship is linear on a log scale. So if you were to put it on a linear scale, it would be curvilinear. It would be hyperbolic, which means that at some point, the production of toxin slows down, right, levels off, regardless of how many cells are ingested, which makes sense because there's got to be a limit to how much toxin can be produced. But the point is, though, that the response is essentially like a dose response, right? The higher the threat, the stronger the response until the limit is reached. So all of these things are consistent with the idea that the prey also adapts to the threat of the predator. Okay, so what do we have so far? We have this idea of this co-adaptation between the predator and the prey, right? Antagonistic relationship. And then the question is, well, can this go on forever? Or is this arms race constrained by costs? Just like in the case of the arms races between the Russians and the Americans. So is there any evidence that adapting to the prey incurs a fitness cost? So we've done this, and some of this stuff is technical. So I picked the simplest example, right? So this is an experiment we did with my former postdoc, Dave Avery. And so what we did is, when there is a cause, right, the idea is, okay, you've now adapted to this stress. If there is a cause to it, the minute you remove the stress, right, then your performance will be worse. If there's some sort of, say, biochemical cost to adapting to it, right, once the stress is gone, you don't perform nearly as well, all right? So we did this experiment where we said, okay, we take a toxin-tolerant predator. So we had grown this predator in the presence of the toxic food for a long time. And then we said, okay, let's do this. In the first generation, we split them in two groups. One gets to eat the toxic prey, and the other eats the safe prey, the good food, right? And we do that. We continue to do that for several generations. We did it for 3 generations. And then every generation, we compare the performance in the toxic food and in the safe food. And what do you see? In the first generation, right, the upper curve represents the survivorship under the toxic prey. And the lower curve represents the survivorship under the control prey or the safe prey, right? And you can see that early on, indeed, the predator does very well with the toxic food. That's better than actually eating the safe prey. But by the 3rd generation, there's no difference. And the reason for that is because what you've done now, if you've cooled off the individuals, right, in the population that incur a cost, right? Those guys are now out of the gene pool. If it's costly, they don't reproduce as well, right? So now you've eliminated those differences right there. So that's one piece of evidence. We have other pieces of evidence, but this is probably the simplest one. Okay, what about the cost of the prey? Is there a cost to the prey? Again, the prey adapts to the predator, but it cranks up the toxin production. If there's no cost, then it would pay to produce toxin forever, right? If there is a cost, then the toxin production is constrained. So a prey, right, like every individual, right, depends on resources, and those resources then are allocated to a bunch of different functions, right? You have to grow, you have to reproduce, you have to store stuff, you have to fight disease, right? Okay, and typically in prey that produce toxin, you could have what is background levels. The toxin is always there, right? But remember that in the case of Alexander, in the prey we study, it's inducible. You bring the predator and the toxin production cranks up. So what is the consequence then of adding the predator, right? If the toxin production goes up, will the other factors go down? All right, that would be the cost there. So we did, guess what? What did we do? An experiment. Because we like to do experiments. Okay. So we did, said, let's do an experiment. Let's have a little fun. Let's do an experiment, right? So this is my current student who's going to graduate in a couple of weeks. And Kihon Park, and said, okay, Kihon, let's do this experiment where we simulate a bloom of the prey, right? And then we're going to look at toxin production and cell growth in different portions of the bloom. And remember that resources are changing over here, right? These guys are resource depleted. These guys are not, right, as they grow. And we do, at different portions of the bloom, we pick them up, and then we expose them to the predator, like we did before. We measure the toxin content and the growth in these cells here, which are with the predator. Those are direct induction. And these folks here, which are separated from the predator, but receive the influence of the predator. So that's the indirect induction. And then, of course, always compared to a control. Okay. So what do we find? Right? So remember the growth, the growth phases, right, are early exponential, late exponential, and post-stationary, right, at the end of the bloom. That's early in the bloom, late in the bloom, later in the bloom, right? And here we have, remember, you have D is the direct, I is the indirect, and C is the control. So in terms of toxin content, we do see that the toxicity decreases as time goes on because there are fewer resources, right? But there's a big difference in the toxicity of the fellows that have been exposed directly to the predators versus those who are just being threatened and those who have not, right? So there's this big gradient in toxicity here, and then there's a gradient in toxicity as we go this way. So there's a predator effect, there's a growth phase effect, and of course there's an interaction effect. If you now look at the growth rate, the cell division rate, right, what you see is the opposite. These guys who produce lots of toxins now have the lowest growth rate, right, and so forth. But there's a problem here because in this case here, these guys were exposed to the predator, so they're also being eaten. So this is not necessarily the best measurement because now you're conflating two effects here. You're conflating the losses due to herbivory, right? And you're conflating the possible losses that are incurring cell growth by producing toxins. So we got to do better than that. All right. So what do we know? We know that the fence is inducible, that it increases with the threat, right? There's a hint of a trade-off, meaning that if the toxin production goes up, the cell growth goes down, right? But we still have to separate the tax that is incurred by each cell by cranking up toxin. So the tax on the individual growth versus the losses that the population incurs due to herbivory, all right? And so if you want to write it as an equation, you would write this: a change of growth is the cell division rate minus the tax that is incurred by these cells, right? Minus the losses due to the grazers, or the predators in this case, okay? So the challenge is to separate this from this here. So that's where molecular biology comes in handy, right? So you said, okay, if we could separate, then this is what we should see. The first thing is that we should see an upregulation of toxin production at the cellular level, and a downregulation of cell growth, right? Then you should see that trade-off between cell growth and toxin production, right? And from there, we think we can figure out what the tax is. Oh, did I just? Okay. So what I think I screwed up and lost one slide. So let me explain it to you. So to separate these losses from herbivory to the losses due to the tax, we said, okay, why don't we just do something that does away with the grazing, right? Let's look at it directly at the cellular level. So what we can do is we can look at gene expression of toxin production and gene expression of cell growth. So you have to find the right markers, and we found them, all right? And whether or not the cells are being eaten or not, you should see, right, if they're being upregulated for toxin production and downregulated for growth. So that's what we did.
Sorry.
Yeah.
It's a very simple question.
Yeah.
Is there any insight on how the algae know it's a predator? It's kind of like,
Yeah.
how,
No, okay, so the answer is no. Okay, so that's not true. So there are ceromons. They can detect a ceromon. I didn't show this, but so in those indirect ones where they don't eat, right? You can see that's, and if you see the increase, that's the indication of a ceromon. And there's a group in Sweden that has actually identified these metabolites that the algae that are produced by the predator that the algae detect. But it's not clear exactly what the algae do, right? And there are many ideas out there that's like quorum sensing, kind of like bacteria do, right? It's, oh, there is a threat here. Hello, threat. And then the other one said, oh, she says there's a threat, better do something, and everybody starts doing that. But that has not been tested. So, okay, so to do this, I said, let's do an experiment. So we did an experiment just like I showed you earlier, right? So where we looked at cell numbers in the treatment and the controls and toxin production, right? So what do you see again? In the treatment, which is where the Copepod is, you see that the cell numbers don't change very much, which suggests that there is a balance between losses and growth, right? Whereas where there's no Copepod, they grow happily, right? And then in the control, the cells don't change their toxicity, but in the treatment, they crank up. So far, so good. I said, okay, so what is this gene expression showing us? Oh, there it is. In the last minute, I changed things and screwed up. But that was the idea between the genetic markers, okay, the molecular markers. Okay, so what do we see? So these three panels are precursors, gene precursors of saxitoxin production. Different forms, okay? So saxitoxin A, saxitoxin A4, saxitoxin B, they're all sort of varieties of the same thing. But in all of them, you see the same pattern, which is the production of saxitoxin cranks up at the molecular level. It makes sense in the treatment, in the presence of the Copepod, but not in the control. Whereas the markers for cell growth here, you see the opposite. There is these guys do well. This is a treatment, right? And there's downregulation of cell growth here. Okay. So consistent with that, with the hypothesis. And you do see, if you now plot those versus each other, you see the toxin production rate on this axis, and the gene expression of cell growth, and you see this negative relationship. So that's the indication of the trade-off. Okay. So it is indeed the fact that it is at the cellular level that if we produce more toxin, we can grow as well. Okay, so how do you get the tax? Okay, so now remember, from this experiment here, that's the tax right there, right? This is the reduction in cell growth that is linked to the production of toxin, right? So we know, and we can use that because if we look at just in the control where there's no predators, right? We look at the gene expression of cell growth versus the measured cell growth, there is a correlation. There is a strong correlation. We could do better. We will do better. This is not published. Yeah, we'll clean this figure a little bit before we publish, right? So you can define the tax as being simply the ratio right here. 1 minus the ratio is going to give you the tax. And then, if you go back to that experiment, right, where we put them in the bottle, copepods, no copepods, and we calculate the tax, and this line here is what the tax would be like. In the typical experiment, right, where you have the two of them and you don't separate them, it would appear that this difference from the two curves is all due to losses due to herbivory, but that's really not the case. So in fact, the tax is significant. It's about 20 to 40% or so, right? Meaning that these animals, right, are going to incur a fitness cost just by the fact that they're producing toxin, independent of mortality due to the predator, right? And in fact, you can see if you said, okay, what would be the optimal situation? The optimal situation is that this is really all I need to produce, right? They're essentially going crazy, right? Which is what the Russians and the Americans did in the arms race, right? Because we had so many bombs, we could have destroyed each other hundreds of times. We could have blown up the world hundreds of times, but we just kept escalating the arms race. So just like that, evolution doesn't optimize. It just is. It is what it is. Okay, that's the problem. Okay, so in conclusions, so we do indeed have an arms race between predators and prey in the ocean. I posit that that's the case, right? That there is co-adaptation from the predator point of view. It's evidenced by the increased tolerance with historical exposure. That tolerance correlates to toxin level in the prey. From the point of view of the prey, we do have inducible toxin production, which is a defense mechanism. And that the degree of induction also depends on the threat level. But adaptation is costly, right? Both for the predator and the prey. And therefore, that's what constrains the arms races, so there is not runaway escalation. At some point, it's just, that's enough, right? Even when they go a little bit crazy, at some point, it's that that's enough. So now think about, okay, so could we have a super bug? Could we have a super copepod or a super clam that would take care of the toxic algae because they can eat it and they don't get sick? That would be nice, but the answer is no. Because what happens when the toxic algae go away? There's a fitness cost, right? And then the genotypes in the population that are non-resistant or non-tolerant, they take over. So we reset the clock all the time, right? So you got this reshuffling of genotypes in the population. And then the next time they come over, those few genotypes that were hanging out, they will come back. And then when they'll become dominant, and then when the blooms go away, the less tolerant genotypes will be there. So there's no magic cure, right? There's no magic cure. The best thing is don't eat the clams. That's my advice. All right. Thank you very much for your attention. I'll take any questions you have. Yeah. Oh, here. So we need to use a microphone because this has been recorded. Extra points if you can bebop. Is that
All right. Okay. My question is a little bit more relating to the first part of your presentation. You were talking, you had the graph up there with the map from the 1970s compared to 2015. And have there been any other studies done in other parts of the world that show the continuation of this trend compared to the north and south, or any other similar experiments?
So for other toxins, that has, did everybody hear the question? Well, yeah. So there are other toxins, right? So there are diatoms that produce a toxin that makes you forget stuff. Amnesia, gives you amnesia, right? The same thing has been shown, right? So these blooms of this diatom that produce amnesia are proliferating. There are a different kind of toxin called brevetoxin that does the opposite. Instead of blocking the sodium channels, it keeps them open all the time, so it makes you really spastic, right? That's really bad news for you. I mean, it gives you a heart attack, that kind of stuff. So all these blooms are proliferating. And the question is, are they really proliferating, or is it that we're getting better at monitoring them? That's clearly one case. Nowadays, we sample them more, right? The other thing is, we are fertilizing the ocean, right? We are putting a lot of nutrients in the ocean, in the coastal ocean. So that's a possibility, is that we're really feeding the, it's like in the lawn, right? And these things can take off. Another possibility is that there's a theory, and the guys who study this stuff usually don't talk about this. there's a theory called kill the winner, right? So this species that engage in defense, because of the trade-off, they don't grow very fast. So they tend to be outcompeted by other species, right? At the same time, another form of adapting to the toxic prey is not to eat them. To learn, this thing is dangerous, I'm not going to eat it. Or when you eat it, you feel the symptoms and you regurgitate the food, right? So the idea is then that other species outcompete the toxic food, but then they're eaten by the predator, right? So the winner of the competition race is actually killed by the predator, and then that allows this toxic prey to increase. So that's another theory that is out there for why they're proliferating. Okay.
Thank you for the great talk. Two questions.
Yeah.
One is if the tax depends on the geographic location of the prey and the predator. The second one is like, how can you keep separated these two measures of fitness, like survival and the egg production? Aren't they related? So if more die, there's less. I mean, why do you keep them as they were independent?
Okay, so there are two questions is whether the tax depends on the geography. And the answer is we don't know. We just finished those experiments. So we just learned how this tax works, right? So that's the next question. So is there a gradient in the cost of the... It's time to go. Bye-bye. Is there a gradient? Okay. Second is why measure both reproduction? Well, they're both important to fitness, right? So you can imagine that a population that have lots of very fecund females, but very few that survive to adulthood, then they're not going to do as well. So you have to consider both the survivorship and the fecundity. So, and then if you know a little, so that lambda measurement, if you can calculate it with linear algebra, you do a matrix where you have fecundity on one end and survival on another end, and the solution of the matrix, the eigenvalue, is the fitness. So that's a quantitative measurement of fitness.
How do you quantify the levels of the neurotoxin?
Oh, good question. So the neurotoxin can be measured by high performance liquid chromatography. There are other assays, but that's what we do. That's what we. In the old days, they would inject a mouse and see how much toxin it took to kill the mouse. So there used to be called mouse units. Yeah, that's too Mickey Mouse. I don't know. Okay.
How many bad clams will do you in? Because I read on Wikipedia just now that less than 1 milligram can kill a person.
Yeah.
How much is in a clam? I mean.
Good question.
Yeah.
I mean, so a clam, right? So if you want to get rid of eutrophication, another way to deal with it is put a lot of clams out there, clams and oysters, because they filter a tremendous amount of water. Well, okay, so we didn't talk about this. So that's a different talk, the mechanism of resistance, right? So the clams that survive, right, have a mutation in the sodium channel. It's a very simple mutation. It's one amino acid changes the configuration of the channel, and then the toxin cannot bind to the channel, right? And you see the same issue, right? In places where these blooms have been around, this mutation is very widespread, right? We thought, okay, we can do that. And I had a PhD student do that. The mutations had never been looked at in copepods. It took a lot of work to do that, and a lot of neurophysiology and so forth. And the answer is, that's not it. That's not it. There's a different mechanism in copepods. And what we think is that we think the mechanism is actually in digestive enzymes that reduce the assimilation efficiency on the toxin. So, you know, even though the toxin is there, you assimilate very little and you reduce the damage to tolerance.
Is there a specific reason as to why the toxic blooms happened at the time they did between the 70s and now, like, as opposed to prior than that?
Well, okay, so the answer is because there was very little monitoring before, we don't know. Again, we know that these blooms have been around from historical records. This is another factoid for your party later. When the colonists came to America, the Indians told them, do not eat clams when there are bioluminescence in the water. So that was empirical knowledge. And the reason for that is because these dinoflagellates bioluminesce. So if the dinoflagellates are there, it's a good indication not to eat the clams, right? This particular, okay. The other is, you've heard this refrain, this saying, don't eat shellfish in months with an R, right? Oh, wait. Without an R. So May, June, July, and August, right? The reason for that is the season when these guys are around. That's the other season. So and the question is why do they bloom? Okay. Again, remember these guys are the competitive, the inferior competitors, right? The dominant competitors are diatoms. Right? But diatoms have a problem. They can't swim. So when the water column becomes warm at the surface, right, it sets up a two-layer system of density, right? So it's light water at the top and cold water at the bottom. And then the diatoms will sink. Once they go past that barrier, they can't make it back unless there's a lot of mixing, right? So what happens is in the warm months here, in the summertime, that's when we get this strong density gradient. The diatoms sink, and these guys can take over. The other thing is, because they can swim, they do a neat trick, which is they can swim up and down the water column. The nutrients tend to be in deeper waters, right? So part of the day, they go down, and they fill up with nutrients, and then that's usually at night, and then during the daytime, they come back to the surface and photosynthesize. These guys have been around for a long time. Dinoflagellates have invented all kinds of tricks to stick around. Yeah.
Anybody else before we? All right, thank you. Thank you, Mark.
Thank you very much. You're welcome. Thank you. Hope you enjoy.